Okay, so welcome everyone, especially those of you who just joined us today or maybe weren't on the boat. My name's Dennis Burton. I'm from the Scripps Research Institute in La Jolla, California. The morning session today is focused on, giving it the title, Antibody Multivalency. My name's Dennis Burton. A series of talks on that topic, opened up by Simone Oostindie. Where is Simone? Should be here somewhere. Here, come please. Simone is the Director of Research and Discovery at Gaius, a company focused on creating a multi-specific antibody platform to discover and develop antibody therapeutics. And the title should be up here somewhere: Avidity Engineering, the Next Frontier in the Development of Differentiating Antibody Therapeutics. Simone.
Thank you. Is here? Thank you for the nice introduction, Dennis. And I'd also like to thank the organizers of this conference, Antonio and Brigitte and team, for this very nice opportunity for me to speak here. It's a beautiful place, and I was actually going to say that unfortunately you're not able to be distracted by the nice views on the boat, but here we are at a very nice place, having again nice views, but still I hope to be able to give you an inspiring talk today about the concept of avidity, which I believe represents the next frontier in the development of next generation biotherapeutic antibodies. And I'd actually like to start off with a slide that was shown by Dennis Burton yesterday. You already saw it on the boat, some of you didn't, but this is actually a very interesting slide because he alongside explained it, that antibodies, the functional activity of antibodies is actually not really dependent on conformational changes, but rather are dependent on avidity interactions. And I think that is very nicely reflected in how the antibody landscape is developing today. Because we now see that there are over 1,000 antibody therapeutic clinical candidates in clinical trials, and over 100 approved. And of those many antibodies in research, still many of them are of the canonical IgG format, so just regular monoclonals. But we do increasingly see a shift towards potentiated formats, and these can be bispecifics, but also multispecific antibodies. And I think these antibodies really take advantage of this avidity concepts that I would like to explain to you today. But we do increasingly see a shift towards I hear that I'm being repeated, but I'll continue. So, I would like to explain the concept of antibody avidity to you alongside a review that we've published last year together with Paul Parren, Janine Schuurman, also both here today, and also our colleague at Genentech, Greg Lazar. And actually, together we decided, so let's have a look at what's out there now in terms of tuning avidity interactions regarding antibodies. And if we then look back at a natural antibody immune response, how can we then fit that in? And how can we explain that based on just an overall concept? And that's what I would like to go into with today. And if you're interested in some further reading about this, please go ahead and read this article. So to start off, when writing this review, we thought, okay, how does an antibody functional response actually look like? And what role does avidity play in this functional antibody response? And we actually came up with this graph actually showing that we think that antibody functional responses are really dependent on three distinct key activation phases. It's not just a on-off reaction. It's a gradual increase that is really distinctly separated into three tiers. And all of these three tiers of activation phases are really dependent on avidity. And to get you all on the same page, avidity, here we refer to that as the accumulated binding strength of multiple affinities that are contributed by individual covalent interactions and cross-linking events. And I think it's very important to stress that these distinct activation phases are separately discussed, because I think all of these phases can be individually manipulated in some way to create more potentiated antibody therapeutics. And I'm even later going to show that we can also combine multiple engineering efforts to increase multiple of these phases. So then what am I talking about if I'm talking about three tiers of avidity? It might seem unknown. So the first phase I'm going to talk about is the zero order scanning phase, which is actually the scanning of antibody over antigen. So it's the binding and rebinding, it's letting go, but it's not actually stably binding to a surface. Then the first important tier is the first stage, that is actually the bivalent or monovalent binding of an antibody to an antigen, so the complex formation of those two. And then the second tier is actually enabled by contacts between antibodies, and those contacts are possibly either Fab-Fab interactions, but also FC-FC contacts. And it's important to stress that both of these contacts actually allow then for interaction with immune effector molecules, which is then the third phase of activation. So immune effector molecules in this case could be Fc receptors to initiate CD or ADCC or ADCP, but also interaction with complements, component C1q. And lastly, I'd like to highlight that really these interactions with immune effector molecules are really dependent on the density of the antibody's binding. So a certain density is required before you reach a threshold to trigger those effector functions, so to trigger third order avidity. And we can then individually manipulate those tiers to increase these natural antibody effector functions. And I'm going to highlight some examples later. So then going back a bit to these densities, so to illustrate, antibody-dependent cellular cytotoxicity or phagocytosis or complement, they're all key mechanisms in a natural antibody functional response to eliminate pathogens or eliminate cancer cells. And as said, they all require distinct immobilized antibody binding to the cell surface before they are reaching this threshold for activation. And I'd like to highlight this a bit. So for example, for ADCC, you would require lower densities of antibodies to bind to initiate this effector function. But for CDC, you would require a bit of a higher binding density to get to this. And also, it's really important to stress that there are also allosteric and intermolecular cooperativity that's really important for these effector functions to be initiated, because you can see that once you get increasing densities, you're enabling, for example, FC-FC contacts. And in the case of complement, those are very important because FC-FC contacts facilitate ordered hexamer formation, which is required for complement activation. But also monovalent binding could facilitate more enhanced ADCC and ADCP. So it's really the density that determines the threshold for activation. So and then staying with the complement activation part for a bit, I told you that increasing densities may enable for monovalent binding and that FC-FC interactions are really required for complement activation. And we now know for quite some time that it's really the ordered hexameric structure, so the antibody bound to antigen in an ordered hexameric ring that allows for the binding of the first component C1q. And Ugular et al. Really nicely solved the high resolution structure of this complex of C1q and C1r, C1s in complex with the IgG FC. So you can really see that it's a nice docking platform for C1q to bind and activate complement. And this is really showing that second-order avidity interactions, FC-FC interactions, really provide the opportunity for third-order avidity to occur, so C1q to bind. So, and then at a later stage, it was really nicely shown by Strasser and team that actually the second-order avidity interactions that are involved in forming this hexamer formation actually occur to two distinct processes. So there's two clustering processes that are involved in this. One of this is solution capture. So the antibody is able to bind bivalently, and via the FC can recruit other antibodies from solution. And then there's also a second process that's actually called lateral diffusion, and that's where antibodies bind monovalently to their target. They also allow for FC-FC interactions more easily, and antibodies can move laterally across the surface to be captured in this hexamer. And it's not only complement that can be activated via this process, but it's also it was later shown that also outside-in signaling is possible by getting these antibody antigens in this complex. So then, up till now, I've showed you what we think consists an antibody functional response naturally, and how these distinct activation phases result in potentiated or potent effector functions. And there are a lot of examples of engineering that you can do to enhance these distinct tiers. And I'd like to go through some examples. And I think one really obvious one that you all know about is actually tuning the self-assembly of the FC-FC interactions. So tuning the self-assembly of the antibodies in a complex. And it was shown really nicely by Dieboldar and team that once you mutate the FC-FC interface, so introduce single point mutations there, that many of them actually are capable of enhancing these interactions more strongly, and then potentiate complement activation for multiple antibodies targeting multiple different targets. Also for targets that are not able to induce complement by themselves. And one example is, I'm showing, and I'm trying the pointer here. Is this working? Well, maybe not, but on the below left figure you can see an antibody against CD37 on its own. It's not able to induce complement activation, but with an E430G mutation, it's very potently capable of doing so. And then, as said, outside-in signaling is also possible. So it was also shown for an antibody against the TNF receptor superfamily member DR5, that introducing this E430G mutation could really potentiate the outside-in signaling for this antibody. And I think it's a nice feat of engineering on a second-order avidity level. So then if you take a closer look at what then results in clinical applications. So for the hexabody technology, there are actually 3 different products now in phase 1 clinical trials. It's 2 hexabody molecules, one targeting CD38 and one targeting CD27. And one dual hexabody molecule, and this is actually a biparatopic molecule, a bispecific, targeting 2 different epitopes on CD37, and I'll come back to this one a little later. So besides tuning second-order avidity interactions alone, you could also think of enhancing those via different ways. And this is the nice part about antibody engineering, I think. There are multiple ways going to Rome. So you could think about multi-epitope targeting of different cell surface receptors. You could also think of multi-epitope targeting on a single receptor. And there are actually several examples in the clinic right now, in trials, but some are even approved. One is actually targeting multiple epitopes on Ebola. It's a mixture of 3 afucosylated antibodies, and that's approved. And there are also multiple products that target multiple cell surface receptors, such as the EGFR, HER2, HER3 family members and others. So I think the field is really moving there. And I also would like to highlight one example. This is actually work that we've done on with antibodies targeting two different cell surface receptors, because why would you do that? What is the benefit? What kind of potentiated functional response do you actually get? So we generated antibodies against CD20 and CD37, which are targets on malignant B cells. They're highly co-expressed. For CD20, it's known that there are antibodies that are able to induce complement, but for CD37, it was known that most of them don't. So that's why we started out looking for them and trying to combine them, see if we could potentiate. So in the middle figure, you could see that there's two wild-type IgG1, CD20, and CD37 antibodies. They're not able to induce complement. This is one CD20 that's not able to do that. But in combination, it really potentiates a complement in malignant B cell lines. And we've also been able to show that in the context of antibodies that contain an hexamerization-enhancing mutation, so antibodies that are already capable of inducing complement activation. But even in combination, you could even further potentiate that, and you can consistently see that in assays where we have primary patient samples from non-Hodgkin lymphoma patients. Some of them are naive, some of them are refractory to therapy, but we see that consistently a combination of hexamerization-enhanced CD20 and CD37 antibodies are capable of inducing high complement activation. And these are antibodies containing the E430G mutation. So then we dived a little further into the mechanism behind this, because how does this work? And we looked using confocal microscopy and FRET assays at the co-localization of these antibodies on the cell surface. And CD20 and CD37, they're really highly localized. So the antibodies are really close together on the surface. And we could do that up to a distance of about 10 nanometers using FRET. And in the figure on the bottom right, you can see number 1 and number 2 are actually the CD20 and CD37 antibodies alone in the FRET assays. And we can see a higher signal once we combine them, higher FRET with number 3 and number 4, which is the combination in different settings. And these are just the wild-type antibodies, but we could see similar things with the hexamerization-enhanced variants of those. So co-localization is one thing, but how close, how do they look? We know that with hexamerization enhancing mutations, you get these hexamer clusters. So I won't be able to show you the details, but we've done some mutational studies where we introduce single point mutations in the Fc domain to inhibit self-oligomerization and promote hetero-oligomerization when we combine the antibodies against the two different targets. And in that way, we were able to show that CD20 and CD37 were actually capable of oligomerizing in heterohexamer complexes. So really together in a complex. And we don't know yet how this really looks. So if it's one or the other, alternating or two, but we do know that they're complexing together. And we also know that it goes most efficiently with this hexamerization-enhanced variant. So the illustration on the bottom right. So then, switching gears a little, I've already told you that CD37 antibodies that we generated, they're not capable of inducing complement activation, and combining with CD20 really nicely potentiated this. But we had a whole panel of CD37 antibodies, and what was interesting is that once we started combining those, we saw some of the combinations actually potentiated complement activation together as well, so CD37 alone. And this was actually found, we found out why, because we did an alanine mutant screening where we substituted every amino acid except cysteine for alanine and looked for where our antibodies bound on the CD37 protein. And we found that we're actually targeting two distinct epitopes on the CD37 protein. So this was really an interesting finding. So we had two different clones, clones 010 and 016, and these combinations really nicely potentiated CDC. But of course, we knew that wild-type antibodies can potentiate, but the hexamerization-enhancing mutation mostly could do even better. So we decided to actually generate, a lot of different types of antibodies, either containing hexamerization-enhancing mutations, the combination, but we also decided to make bispecifics. And we did so using a file exchange method, where we actually exchanged the different antibodies against the different epitopes, sometimes introduced a hexamerization-enhancing mutation, and we actually ended up with a bispecific targeting 2 different epitopes on CD37, containing a hexamerization-enhancing mutation. And we decided to test that one alongside all of the other options and combinations that were out there. We tested it on B cells from patients that are really refractory to a lot of lines of therapy, including CD20. And we actually found that this dual hexabody CD37 molecule, so this bispecific with this E430G mutation, really was the most potent in activating complement compared to every other format that we tested. I'm here illustrating 3 others, so the combination of 2 CD37 antibodies against the 2 epitopes and the single ones. And we really consistently saw this across different patient samples. And we decided to screen a whole panel of primary patient samples from CLL patients, non-Hodgkin lymphoma patients, and this is ex vivo, and we did see that we really had a nice dose-dependent activation of CDC, and that we could see consistent complement activation across different indication subtypes, but also from B cells from patients that no longer respond to standard of care, including rituximab. So the right figure shows on the left the efficacy of rituximab in inducing complement. And then on the right, you see the newly diagnosed versus the relapse refractory patients in the context of the dual hexabody CD37, and it's really consistently showing activity. So based on this data set, we decided to move this antibody to phase 1 clinical trials. So then, switching gears again. Up to now, I've told you a lot about the work we did with the hexabody technology, and we also decided to do some further engineering on this one. So we obviously I showed you that by combining 2 antibodies against 2 different cell surface receptors, we found that they could oligomerize in heterohexamer complexes. And this was an interesting thing because, I mean, we hypothesized that maybe we can make this codependent or selective. And what do I mean with that? We actually wanted to make antibody combinations to act as Boolean logic AND gates. In other terms, what we would like to achieve is to have activation only if both antibodies bound to a surface of a cell that co-expresses 2 targets, and then only get hexamerization there, and not get the hexamerization on cells that express either one of those 2 targets. So there are several potential advantages to do this. One of them is potentially having reduced toxicity and increasing a therapeutic index. You can change the dosage. But more importantly, we may also be able to expand the target space to previously inaccessible targets. And another potential advantage may be to avoid the hook effect that we would usually or sometimes see with covalent bispecifics. So what we would like to end up with this technology is a sort of an on-off switch, where you have this heterohexamerization occurring on the cell surfaces of cells co-expressing 2 targets, while no hexamerization occurs on cells expressing 1 target. So how do the molecular design aspects of this technology look like? I'd like to walk you through it. So if you start on the top left, we have the hexamer, and we're achieving this by introducing an E430G mutation, because many targets are sensitive to hexamerization once we introduce this mutation. So that was the first. And then we needed to make it codependent, so we started introducing additional mutations The second one being mutating the FC-FC interface to inhibit self-interactions between antibodies of single targets, but promote interactions between antibodies targeting different targets. And we did so using K439E and S440K. They're really charge dependent, so they're repulsing each other when they're alone, and when together they're forming these heterohexamer complexes. We then still needed to solve one thing, because we could only target or at least we could aim for the effector functions that are dependent on hexamerization. So what we needed to do is silence the hexamerization-independent effector functions, and one of them is, for example, ADCC. So we decided to introduce G236, to mutate the G236 and G237 positions in order to do that. And these two positions are actually at the shared interface of binding to Fc receptors. So what we then ended up with is actually a HexSelect1 component and a HexSelect2 component, either one binding to different cell surface receptors, one containing the K439E mutation, and the other the S440K, to really get them together in co-complexes once together bound on a cell. So then, of course, our aim was to show selective depletion of a particular cell subset within a mixture. And for that, we took a model system. And the model system in this case that we took was actually antibodies generated towards CD20 and CD52. CD20, as you all know, is exclusively expressed on B cells, so this is really a stringent, strict, selective target. But CD52 is not only expressed on B cells, but also on T cells, and it's very well known that there's high toxicity observed when clinically the antibodies against CD52 were tested, and of course that's something that we would like to prevent. So we thought this is a very nice model system, because here we could show that if we make HexSelect antibodies against these two targets, that we may achieve selective depletion of the B cells, but not the T cells. So how did we do that? So we used a whole blood model system where we have different cell subsets there. And on the x-axis you see the presence of the B cells, on the y-axis you see the presence of the T cells. And on the top left is a control antibody showing nicely the presence of both populations. And then our HexSelect1 and HexSelect2 single components should be silent, because we don't want any hexamer formation when they're bound alone to a cell that expresses only one of both targets. And that is actually the case, still those populations are there. But the combination, we actually wanted them to hetero-oligomerize into these complexes and only deplete the B cells expressing CD20 and CD52, while sparing the T cells expressing only CD52. And that was actually the case. You see that the B cells are nicely depleted, and the T cells, they're still there. And then we validated this concept. This is, of course, one human whole blood donor. We validated that using multiple donors, and we consistently see that in the pink characters, that we see nice depletion of the B cells, but not the T cells. And we actually also compared this to our regular hexabody format. So antibody combinations with just an E430G mutation, they would oligomerize just everywhere. Those are actually the black symbols. And you can see you get depletion of the B cells, which is nice, but you also get depletion of the T cells, and that's something we didn't want. So that showed us that this technology could work. And I don't think I have time to go into more details, but we also validated this concept in vivo. So using humanized mice, we injected the combinations, and we actually nicely showed this exact same feature. So depleting B cells in these mice, but not the T cells. And we also we looked in the blood, but also different organs. We also checked complement deposition, and we could nicely show that there was no complement deposition for this combination, but that there could be for the hexabody antibodies that were the counterpart or the control in these experiments. So then, I'd like to end with 2 slides about Gaius, where I'm working now, because I've done a lot of work at GenMap over the past few years, but I decided it's time for a change, and I actually joined Paul Parn and Ton Lochtenberg in March this year at Gaius, and what we aim is actually to build a startup biotechnology company that generates state-of-the-art multi-specific antibodies. And I hope that I've convinced you now that there's a lot of nice biology around playing with avidity interactions at different tiers of activation phases within a natural antibody immune response. And we actually want to unite this biology or this knowledge and translate this into developing multispecifics and, well, exploit opportunities provided by the avidity engineering concept. So that's the idea behind this company. And to highlight a bit about what we're now doing, so what we actually want to do is we would like to generate phage display antibody panels, so really large antibody panels, together with a world-class partner, my role here for Journey Biologics. It's really nice to be able to collaborate with such an excellent team of scientists, so I'm really happy with that. And what we want to do is with these panels really perform unbiased in-format functional screening. So screen thousands of multifunctional antibodies, and I think this is important because we usually start out with a monoclonal antibody, we have panels, we select them, and we have a single lead. And then at a later stage, we say, okay, maybe combining is a good effort, but then you're missing everything that's before, all the panels that you've generated. Maybe if you combine them in a different way, you can get really nice, unique antibodies with unique mechanisms of action and drug-like properties. And I think really starting earlier, screening at an early stage, really allows us to identify these unique mechanisms of action. And well, I hope to be able at some later stage to share a bit more about what we're doing, the programs that we will be running. That's for a later conference, I think. And I'd like to summarize. I think avidity binding plays a central role in antibody functional responses, natural functional responses. We can learn a lot from how antibodies naturally behave in the body. And I think the response kinetics governing those responses can be described by 3 tiers of avidity binding. And each of these tiers provide opportunities for avidity engineering. And what I haven't touched upon so much is that, well, I think it became clear from the talk, but I think individual interactions contributing to avidity or individual interactions, they're weak, but avidity, so combining, clustering, really improves this into nanomolar, picomolar ranges, and strength really is in numbers. And I've really mostly shown you how we can manipulate the CFC context to enhance avidity, but this can also be applied to FAB. And in the review, we described some examples of how FAB-FAB interactions can be manipulated. And also, there was a really nice paper recently by Greg Lazar, also showing an application of FAB-FAB interactions and how they can result in potentiated responses. So to end, yeah, I think really there's a novel frontier that we have to get past, and I think there's many exciting examples at this conference of how we can use avidity to tune, and I'm really excited also for the coming talks. And I'd like to thank you, but also would like to thank a lot of the people that I collaborated with. I don't want to forget those, of course. GenMap, I worked a long time at this company until March. So a lot of people have been involved in this research. It's really a team effort, especially the HexSelect program. It's really, this was really a large effort to engineer. But there are also external collaborators that we worked with, Amsterdam UMC, Genentech, University of Virginia, really the early research targeting CD20 and CD37 in combination. And of course, yeah, thanks also to the 2 people in the front here. Legacy, I think, in the field. And yeah, thank you.